looking good. I'm just going to do a little pointing out here. So what we see here on the slides, on the left-hand side, we see our original chromosomes, right? So the purple and the green together, this is a homologous pair. So each of these is a chromosome. They would have the same genes, but potentially different alleles for those genes. And after replication, what we end up with, sister chromatids within each of those chromosomes. So our sister chromatids, like these two purple legs, right, this one and this one, are going to be identical copies of the same DNA. Same thing within our green chromosome, right? So this is an identical copy of this. But if we take a look at, for example, this purple chromatid and this green chromatid and compare those two, those would be non-sister chromatids. So each of them is a chromatid, but they have different information because they're copies of different chromosomes within the homologous pair. So that's what we're talking about when we're talking about sister versus non-sister chromatids, a little more practicing of our understanding of what a chromosome is. Okay. So we talked a bit about the structure of our chromosomes before. So remember that after replication, we end up with two identical copies of the DNA. Those two copies are still stuck together, basically, at the center. This region called the centromere. We're also going to see a structure today called the centrosome, which is made up of two centrioles. And I actually don't care whether you use the term centrosome or centrioles. Those are equivalent for our purposes here. We're going to see that these are structures that we're going to find at the ends of our cell during mitosis. So here we see a centrosome with its centrioles here and one here. What the centrosomes and centrioles do is they're going to be attaching to something we call the mitotic spindle, which is made up of microtubules, which are basically a kind of protein related to our cytoskeleton. They're going to attach to our chromosomes. We're going to see them attaching to this central part, to this centromere in the replicated chromosomes, so the ones that have sister chromatids within them. They're going to be responsible for basically pulling apart our sister chromatids during mitosis. Okay, so that's one structure we haven't talked about yet, but it's going to be important today. So as we're talking about mitosis, we first need to step back a little bit and think about the life cycle of a cell. So we have this thing called the cell cycle, which is the life cycle of the cell. And it's composed first of two basic parts. We have interphase, and I'm circling interphase here. Right? So interphase is quite long. Most of the time, a cell is in interphase, and during that time period, it's not actively dividing. So it's just out living its normal life. A smaller component of the cell cycle is mitosis, which we'll also refer to as the M phase or mitotic phase. So that's this part, right? We can see in this diagram, it's about a third of the circle. So this is mitosis. This is where we have our named phases, starting with prophase, ending with telophase. Okay, so this is the portion, this M phase is the portion when the cell is actually physically dividing. But before we get to the division, there are things happening in interphase, so the part I circled in yellow, that we want to know about. Okay. So we're focusing right now on interphase. Parts of interphase are basically going to prepare us for cell division. So the truly sort of part where we're not doing much related to mitosis is G1. So during G1 phase, we'll be expressing our genes, we'll be making various proteins, we'll be living our normal cellular life. We're not doing much related to cell division at all. But as we enter the next portion of interphase, this S phase, S stands for synthesis. Synthesis means like making something. So what we're synthesizing during S phase is new copies of DNA. So this is when replication, that stuff we talked about on Wednesday, is happening. S phase of interphase is when we're making our copies of the DNA. So we'd go from a chromosome that looks like this, copy it, right? Pairing up A and T, C and G, ending up with sister chromatids that might look like that. So that's S phase. In G2 phase, our G's stand for gap. So the cell basically rests, prepares more energy for cell division, copies some organelles. So this is kind of like a resting phase before we enter our actual cell division. So here we can see another image of what some of the pieces of our cell cycle look like. So our cell cycle begins here. All the way here in this image, we can see interphase, right? So all of this part is interphase. So we can see the cell growing. We can see it kind of deciding whether it wants to divide. And if it does, it enters S phase, the synthesis part of interphase. So we can see that its chromosome here is now replicated when we get to S phase. So if we looked at the cell during G2, it would also have that kind of X structure. And then eventually it will go into cell division. Now we're going to look at this image on the right. So this image on the right is tracking us through what's happening with the chromosomes. So it's showing us G1 and S phase, comparing those parts of interphase. I'm just going to put an I here, reminding us it's interphase. You can write out the word. So here we see two homologous pairs. So we have our homologous pair chromosome A. We got a big version and a little version. We have our second homologous pair. We got a big version and a little version of chromosome B. And we can see that these chromosomes look kind of stick-like. It's because they're just one copy of the DNA. During S phase, this synthesis phase, we can see that we create the sister chromatids, so we duplicate the DNA. So if we now focus on this big A chromosome, we can see it's got this X structure because it has two copies of the DNA within it. So it's got these sister chromatids inside. Then this image is also showing us some really important parts of mitosis itself. So it's not showing us all the phases, but it is showing us the parts where we're going to have really significant changes to the arrangement of our chromosomes. So here we're tracing as well the chromosomes in metaphase of mitosis. So we can see all the chromosomes lining up one by one in the center, right? So they still have this X structure like they did in S phase of interphase, but now they're lined up at the center of the cell. And we can see how they're connected, right, to our centrosome and these microtubules. This stuff is what we call the mitotic spindle. It's getting ready to pull them apart. We can also see what we call the daughter cells at the end of this process. So in telophase, we actually create two cells from this one cell. And we can see that these cells at the end look just like our cells at the beginning up in G1, right? So they now have that unduplicated material, just have an identical copy of this big A, identical copy of this little a. So this is the purpose of mitosis, is to just make identical copies of this cell. I just want to highlight for us here, this first cell that we start out with, we refer to as our parent cell. Parent. All right, writing with a mouse is not super useful, but that's our parent cell. We're also going to note that it's a diploid cell. There we go. The way we know it's a diploid cell is because it has homologous pairs, right? So it has a big A and a little a. That's how we know it's diploid. So if we look down at the bottom, right, at our daughter cells, right, our daughter cell, we'd see that this daughter cell, also diploid, right, has big A, little a again, just like the parent cell. So it's also a diploid cell. So if you were to compare the colors and everything, we would see that the parent cell is genetically identical to the daughter cell. We see the exact same picture. You could just copy paste. So this is the basic process of mitosis. Now, mitosis happens in cells we call somatic cells. Anytime you see this root in the future, soma, just so you know, this refers to like a body. So what we mean here is that a somatic cell would be like a heart cell, a lung cell, a cell in your big toe. The reason we're emphasizing it here is when we get to meiosis, which is a similar process but different in some really crucial ways. Meiosis is going to happen very specifically in germline cells, which are the cells that go on to make sperm and egg cells, basically. So mitosis is happening everywhere in your body except for those germline cells for sexual reproduction. So it's necessary for tissue and organ growth and maintenance, right? If you get a cut and you need new skin cells, you could do mitosis. Or if you're a little baby, you want to get bigger, you do mitosis. So it's just a copy machine making identical copies of our cells. So here we see the chromosomes, again, circled out here, starting with this homologous pair, G1 phase. We replicate them during S phase, creating sister chromatids. And then after mitosis, those sister chromatids got pulled apart. They're in separate cells, leaving us with two daughter cells that are identical to our original cell. So for our purposes, because this is a big flow process, sometimes you'll see slightly different terminology for the phases. So I'm just highlighting for us here. The phases we're going to care about are at the top. We're going to call it prophase, metaphase, anaphase, and telophase are the ones that we're thinking about. So we're going to run through those real quick. Okay. So this image starts us off with interphase, and specifically G2 of interphase. So this part in brown is not directly part of mitosis. It sets us up for mitosis, but it is not itself mitosis. Part of the reason we can see that is that we have our chromatin, our genetic information it's duplicated, but it's still kind of messy. We're going to see as we enter mitosis, we bundle up and wind those chromosomes nice and tight so that they're visible and identifiable. And we can see that our centrosomes, they're not really significant at this point. It's not totally ready for division yet. Okay. So prophase is going to be our first part of mitosis. So what we should note here are a couple things. One is that these centrosomes, right, these structures, are starting to pull apart and they're starting to move towards opposite ends of the cell. And they're forming this spindle between them. This is referred to as the mitotic spindle, right? So this kind of cage structure, or we can think of them as fishing lines. Our mitotic spindle is beginning to form. So that's one thing that's happening in prophase. We want to track three things. Other thing that's happening is that our chromosomes are condensing. So we can now see that these chromosomes, they're not messy like in G2 over here. They are condensed into these X structures, so we can see them. The other thing we're going to see starting to happen in prophase, and for our purposes, we're basically grouping this prophase and prometaphase as just one thing. So the other thing that's happening is that the nuclear membrane is starting to dissolve. So if you compare the image in this blue section, prophase, and this image in green, the prometaphase, you'll note that the second image doesn't have a nuclear envelope. So remember that the role of the nucleus and of that membrane, that double membrane surrounding it, the nuclear envelope, that's to contain the DNA, keep it all in the same place, make sure it's not getting messed with. As we start to do cell division, we do want to mess with that DNA. We want to pull it apart, and we want to put it into two whole new cells. We wouldn't be able to do that if it was still protected by that membrane. So that membrane has to dissolve in order for us to actually distribute those chromosomes to two new cells. Okay. So our three things are our mitotic spindle is forming, the chromosomes are condensing, so they're winding tighter, and the nucleus is dissolving. So that takes us into our next phase, which is metaphase. So what you see in this image, right? Over here, we had our first, what the images from the last slide, but now we're zooming in on the next three steps. Okay. So during metaphase, the important thing we have happening is our chromosomes lining up one by one down the center. And we can see that the position of the centrosomes and this mitotic spindle, we have centrosomes on either end of the cell. We refer to the ends of the cell as the poles. So just like you have we have a north pole, south pole on Earth, we've got poles on our cell now. And we have these microtubules attaching in to our chromosomes here. So that's metaphase. This central area where we have those chromosomes lining up, that's our metaphase plate. Right? So just like you shoved a plate down the center of the cell here. Next step is anaphase. So in anaphase, what we have happening, right, is this mitotic spindle, right? We had these spindle fibers. They're now starting to contract. So these microtubules, they're getting shorter. And the effect is that they're pulling the things they're attached to towards the poles of the cell. So they're pulling from this centromere area in the center of our sister chromatids towards either end of the cell. So we're basically ripping apart this X structure. So we have one sister chromatid going towards this north part of the cell, the other sister chromatid going towards the south part of the cell. Now that's important because in our daughter cells, each of these sister chromatids becomes its very own chromosome. So that's what we see here happening in telophase, our last phase. In telophase, we see that we now have those individual, or I should say those sister chromatids are now their whole own chromosome. They're still in homologous pairs, but they no longer have that X structure. We see the nuclear envelope reforming, right? So we'd start to reform that membrane around our genetic information. We'd start to see that spindle disappearing. So basically telophase is just the opposite of what was happening for us in prophase. Everything's kind of running in reverse there. Okay. So this is a dynamic process. And if you were to look at it using a really good microscope and some dyes, what you'd see is that the microtubes actually kind of go fishing for the chromatids. So they're trying to latch on in order to pull them apart. And I believe we